My name is John Quigley. I'm the Assistant Dean for Advancement for the College of Arts and Sciences. I want to welcome you to our presentation today, The Future is Alive, Why Physicists Are Studying Biology and How It Could Change the World. So right now it's my pleasure to introduce Karin Ruland. Professor Ruland is Dean of the College of Arts and Sciences. Karin, it's all yours.
Good morning to some of you, or good afternoon to some others, and thank you for joining us today. I'm happy to see quite a few familiar faces. As John just said, I'm Karin Ruland, the Dean of the College of Arts and Sciences. And I'm also a distinguished professor of chemistry here at SU. So just as with you all, it has been an absolutely surreal few months for me, both as a person and as a dean. But I'm happy to note that we successfully finished the spring semester online and that we're truly looking forward to welcoming back our students in August. Speaking of things I'm excited about, today's presentation on nanoscale machines is a real great one. In a moment, you'll hear from Professor Jennifer Ross, a new member of our prestigious physics department and a rising star in her field. But first, let me tell you a little bit more about her. Professor Ross holds a PhD in physics from UC Santa Barbara and was named a Fellow of the American Physical Society in 2018. A highly regarded research scientist, educator, and leader, she came to us last year from UMass Amherst, and we're so happy to have her here now as a part of our absolutely stellar physics department. Already, she's making a difference on campus and off. Before we transitioned to remote learning, she had brought her students to the dome for hands-on memorable lessons in air pressure before its inflatable roof was gone for good. And when we all left campus in March due to COVID, she rallied her colleagues and students in a face mask making effort. And as for many other faculty, her research goes on. I'm excited and honored to share it with you. The work of Professor Ross and her colleagues is truly the stuff of science fiction. It is innovative in bringing a physics perspective to bear on biological systems in order to imagine innovations such as self-building houses or self-repairing streets. Sounds pretty incredible, right? But that's just the type of thing we're tackling here at the college. And frankly, I can't think of a time when we've needed more to hear about some encouraging news on what's going on the scientific horizon. So without further ado, let me please welcome today's wonderful scientist and presenter, Jennifer Ross. Jen?
Thank you so much, Dean Runland. I really appreciate the introduction. I am going to share my screen now so that you can all see the slides that I've created for us. And I do want to say thank you so much to the Dean and everybody else for welcoming here at Syracuse University. It is really fantastic to join this fabulous department and college. Just a few of the things listed here that the Dean had mentioned. One of the things I'm most excited about over the awards that I've won is the Cottrell Scholar Award, which is a researcher and teaching award. So I know that for me, doing teaching is really important. Working with students is really important. And it was really the hardest part about having to go remote was not being able to see them anymore and have them in my lab. So without further ado, I'll just get started. So as the Dean alluded to, I want you to just sit back and think a little bit about the world of the future. I am very excited and motivated by science fiction. I want you to imagine a world where bridges, roads, dams, pipelines will heal themselves before you have major environmental problems or, burst dams like we just had in Michigan recently. The bridge I'm showing is the Tappan Zee Bridge, the longest bridge in North America. So all of these infrastructures we are constantly using and constantly repairing. It'd be amazing if in the future we actually had materials that could sense when they're having fatigue and fragility and actually either signal out to us to let us know before the dam breaks, or even better, have the ability to heal itself. So much like your skin can cause a scab and heal itself, we would love to create new materials that have properties of living systems but aren't actually alive in order to achieve these types of things. And I want you to also imagine a world where you could have personalized, instantaneous diagnosis and treatments of disease, because you now have nanoscale machines that we've created that can live inside your body and constantly be reporting out to your doctor about your current state of health. Right now, one of the problems is you feel bad. I don't know if this happens to you, but this happens to me. I feel bad, or my children are feeling bad. I take them to the doctor, and as soon as they get to the doctor, they're bouncing off the walls like they had nothing wrong with them, right? So the idea that you could be consistently monitoring what's going on in your body, but continuing to talk to the doctors throughout the time, this could come from new nanoscale technologies that are based on biological technologies. One that's really hitting close to home right now is, you can imagine a world where fashion turns into protective equipment. So you could have a set of clothing where you do not need to know that there is a toxin or a pathogen nearby, but it itself changes its porosity automatically when it detects a toxin or a pathogen nearby, closing itself up and creating protective equipment that you're just wearing all the time. This would require new materials that are built into fabrics and cloths, which are a type of material we interact with every day. This would also help our military in going into situations that may be problematic or have chemical weapons present. And so all of these things are ways that we can imagine a new world that is based on novel materials that we don't yet have, but we are at the very beginning of understanding how these could work. Biology is what is inspiring us. Biology has solved so many of these problems already. So I'm showing you 3 movies. They're from the molecular to the cellular to the large scale, human scale. And in the first one, what you're looking at, and it's going to replay in a second, it's a dish of cells where you can actually scratch the cells to create a wound. And these cells are able to grow into the wound and heal up the wound. This is a system that we use in the laboratory, but it actually teaches us about things like, how would you create a material, a tissue is a type of material, that can heal itself. On the far right, I'm showing you a single cell that is going through the process of cell division. Creating new material is something that cells and your body can do, but we are not very good at that. It would be so great if in Syracuse potholes could reseal themselves by creating new road material as they're starting to crack and crumble. And then the one in the middle, the movie I'm showing you in the middle, is the mimosa plant. You can sometimes find this around here in this area. It's also called a touch me not plant. And this plant, as you can see, as somebody touches it, there's actually, it's a large scale perturbation or kind of change to the plant, but there's now a molecular scale sensor inside that is actually knows that it's been touched and creates a chemical cascade that causes the whole plant to close. So these kinds of sensing and responding mechanisms are something that biology is already very good at, but we in the laboratory and in industry are not quite there yet. So we want to take these biological ideas and we want to turn them ultimately into new and innovative materials that we can use. And I'll give you first an example that's in the literature. It's not really in the literature, it's in the movies, of a material that might be of the type that we'd like to create. So this is actually a clip from a movie. If you know the movie, write it in the chat, and the people who are helping us out here will hopefully look through it. And I don't know if this is possible, but if you're the first one to catch what movie this is, you can get a gold star. You may have figured it out by now that you've seen 2 of the actors. So this movie, I can't see the chat, so I'm just gonna keep going for it as if somebody has answered in the chat.
We've had quite a few answers, Jenny.
Did you, were any correctness?
Several Terminator 2s and a few Terminators.
Perfect. So yes, now it's very clear that this is Terminator 2. And the reason why I always show this movie is not because at Syracuse we're interested in creating killer robots from the future. We are not interested in that. I just want to make that very clear right now. No killer robots. But we are very interested in autonomous systems and autonomous materials. And so there's many ways for things to be autonomous. Right now, we're often thinking about self-driving cars or drones, but actually we can create materials that have some of the same autonomy, that they can sense, respond, move, self-organize. This material, to me, again, I love sci-fi. I love using it to be inspired. This is a material that has so many properties of a biological material. And yet, in this movie, it doesn't use all of it to its best advantage. In this movie, this material decides to become a police officer. I'm not really sure that was the best choice. To me, it could have easily killed John Connor in a different way. Okay, I'm getting off topic. The point is, this is an inspirational material. If you could create a road that could reconfigure itself, it could heal the cracks and hopefully not kill children. As another example, so this is Exhibit A. What do I mean by this? What's the pipeline of this look like? From being bio-inspired, right, to being all the way to creating new innovative products for everybody to be using and making our world a better place. So I'll just give you an example. Here's a biological system, horses. Horses have driven humans to innovate for millennia. So the first thing we did with horses was harness horses, right? So we could take a biological thing and we can harness it to use it for our own purposes in order to move things around faster. Or if you're like the woman who's pictured here, she actually rescued and would guard stagecoaches in the Old West. Okay, so we can do many things when we harness biology. But harnessing biology only takes you so far. You're still limited by the biology. So the next step is to synthesize it. And when I say synthesize it, I mean turning something biological into something human-made, right? And so the next step from going from horses was to go to horseless carriages or cars. And so this is a car from the 1916s, very early model. You can see how very similar it is to the original wagon with the horse. And it's one of the first ways that we really try to replicate the horse. And once we can replicate it, we can further innovate it. And now we have cars that look nothing like horses. They look nothing like the original wagons. They can do so many things, and we're talking about having them be self-driving very soon. And so we can continue to build on these things, but everything that we're talking about here was bio-inspired in the first case. So we want to create a material instead. So let's use the same kind of process that we use as humans for creating cars from horses to try to understand how we could create a material that has the ability to move and sense and respond. So the first kind of place where physicists have been looking at this is looking at energy and power, right? We need that same thing, but in smaller systems that are biological systems. So what I'm showing you here are swarming bacteria. And so if you watch this, these bacteria really are randomly moving around, but there's so many of them that they actually kind of create these swarms in local aligned areas. This type of system from a physicist's point of view is often called active matter. And we're very interested in studying how we can harness this type of material. So in fact, that has been done. This is an example of a turbine that is being moved by bacteria such as the ones you see on the left. And so the turbine is being rotated because the bacteria have swam into the kind of grooves of the turbine and are now pushing it around. This can now be multiplexed, and now you can get productive work out of a biological system and actually create, when I say work, I really mean forces over distance, right? That's what gives us that work in physics. That's how we define work. And so you're able to actually do this. This is real. This is not a cartoon. This is a movie from a true scientific setup that has been created, and it was created through nanolithography, so nanoscale etching of a hard material to capture this. And this wasn't done at SU, but this is where a lot of us are looking to go into places like this. Once you harness it, then we need to synthesize it, creating a man-made version. And in fact, people have been able to do this. So if you look on the right-hand side, these are now no longer bacteria. They're actually little polystyrene or plastic beads. And these beads have a little bit of metal in them. When you put them in the right chemical conditions and shine light, they actually self-propel. And in this case, they're slightly magnetic, so they want to stick to each other, and then they can create the same types of turbines. So now we've created freestanding windmills. If you can now harness this activity. The next step of this, and many people are working on, is innovating this. So they're creating new versions of the synthetic swimmers that are replicating the bacteria, but they're also doing one more thing. They're trying to miniaturize it. Miniaturizing things is really essential for progress in many different places. So for instance, in electronics, we have miniaturized things throughout history, right? As we've started with computers that first filled a whole room, and now we have this, right? Now we have our phones that are able to do, you can actually, this has more power than the original space shuttle, right? And so trying to minimize this is really important, especially for creating a material. Most materials that you interact with, like this hairbrush, are made from molecules, not from big particles or from bacteria. That doesn't usually make up the basis of the material. You need a molecule to start with. So let's try to figure out how we can minimize it. And this is where my work starts to come in. So inside of bacteria, there are actually other little tiny things that in a sense are little machines, as the dean referred to earlier. They're nanoscale machines, and they're made out of protein. So we all know what protein is. If you buy milk, it's got a lot of protein. Protein's really hot right now in your diet. But it's always been important for us because inside of your body, all of the processes inside are driven by proteins. This protein that we're looking at is an enzyme, and it's actually a tiny little nano machine, and its job is to catalyze chemical reactions. So again, we're now going from the micron scale to the nanoscale, which is the scale we need to be at in order to really create a material that's active. So if we look at this enzyme, it will take in a molecule, and then it will catalyze a chemical reaction and basically turn it into a new molecule. So for those of you who are chemists, you might know the words substrate and product. That is what's happening here. But I'm a physicist, so I like to think of them as fuel and exhaust. And so we know that we can create a lot of energy and a lot of force out of chemical reactions. So for instance, on the right-hand side, this is one of the SpaceX rockets. I did the launch, I didn't actually see, I saw the aborted version of the launch. Okay, so they all got aborted yesterday. But these rockets all launch off of chemical reactions. The space shuttle is just hydrogen and oxygen creating water, but it's an explosive reaction that gives you a lot of propulsive force. So makes it so that you're able to move. So it shouldn't be that surprising that we might be able to create a rocket out of another chemical system. In this case, it's just a biological system. And that's what we're trying to do. The first step to getting here is to check, do these enzymes truly move more when they are having their fuel present? And so in fact, this was a paper that we just published within the last year at Physical Review Letters. I know it doesn't sound fancy, but for physics, this is a pretty fancy journal. And what we did here for the first time ever, using our microscope that can see individual proteins, we actually observed directly that enzymes can diffuse and move faster when in the presence of their fuel. And so what you're seeing on these two movies is the same exact enzyme without any fuel. And so it basically just bobs around, much like an inner tube would bobble around on the surface of a pool, just randomly. But when you add the fuel, you can see with your own eyes, these movies are actually playing back at the same frame rate, that it moves significantly faster. And so this was the first time that it had ever been directly seen that enzyme turnover can move faster. And what we were able to do this because we have the ability in my lab to see individual enzymes one at a time. this is really surprising. When we published this result, many physicists were very, very surprised. Why? Because this directly contradicts what Einstein said in 1905. So Einstein in 1905 one of his three big theories, along with relativity, was about diffusion. So I didn't know if you know, but Einstein was a biophysicist. And so he took data that he had from biologists who were studying how particles move in water, and he came up with a theory that has stood the test of time until today. And what we have found is that if you have an enzyme and it has the ability to perform its reaction, something is different about it where Einstein's theory doesn't work. So you might ask, well, what is the right theory? And here's the exciting thing. We don't know. So this is so exciting that we are actually at the place where we've now seen an experiment. It contradicts an over 100-year theory, and we have no idea how it works. And so our lab is still trying to figure out how this works. So the next steps of this for our lab is to harness it. So we've already basically empirically shown that this works. It's okay that we don't have a theory. We don't need a theory in order to harness things. We don't have a theory of how horses work either. And it's okay. We can still harness them and we can still use them to make synthetic things. So we're going to do that. So the way we're going to harness this is using DNA origami technology. So when you have a bunch of DNA, it'll actually bind to itself to make the double-stranded DNA. And in this case, we can program the sequence so that when it binds to itself, it actually will fold into a really cool structure. We can pick any structure we want, but we're going to start off with a rocket. And so our rocket is just going to be a cylinder of DNA. And now, because we have the DNA, we can also program where on the rocket we have our enzyme fuel. And so our enzymes are going to be at one end of the rocket. And when we turn on the rocket, meaning we add the fuel, we are now going to get our activity, which hopefully will be the propulsion of these rockets. So that's what we're going for. You might think this sounds crazy. The National Science Foundation thought it sounded really cool, and we actually just got a new grant funded. It's a collaboration with two other scientists, and one who's an expert on DNA origami, another one who's another physicist. And we are really excited to study this. We really want to create these things because we really think we can actually not only harness the individuals, but actually harness the swarms and get some information. So we're going to create the same kind of swarms you saw with the bacteria with these rockets, and we're going to harness that to create a material that has the ability to move and reshape and ultimately respond. So the next steps after all of that will be, of course, to synthesize it and innovate it, and I'm so excited to be able to do this at Syracuse University. Here's a picture of my lab with the students that are part of it. Many of these students are actually Syracuse students, and they were all in the lab this January for an extra week of instruction. They didn't get any credit for it. They also didn't pay any money for it, but it's called the Bio Boot Camp. And it's a 1 week experience where they get hands-on training in the lab. When they're done with the Bio Boot Camp, they actually get their own projects. So many of these students are actually working on the project that you just saw that me describe. I'm so excited to be able to work not just with the physics department, I'm also a member of the Bioinspired Institute, which is a multi-college, multi-department institute at Syracuse University. It's really pushing forward a lot of these amazing fundamental ideas. And I'm so proud to be part of this family and to be able to help these students to dream big and be inspired. So that's all I have. I will now stop the share.
Thank you, Jenny. That's really amazing stuff, and I love your enthusiasm and the way you were able to translate that into ways that someone like me can understand. So I appreciate that. So what I'd like to do now is open up to if you have any questions. You can either write your question in the chat and I will convey it to Professor Ross, or if you want to raise your hand by clicking on the button to do that, I will call on you and ask you to unmute so you can speak your question. So, so we have one question that just came in on the chat from Barbara, and she's wondering, are the students in the photo you showed physics students, biology students? What are they studying? Where do they come from?
That's great. That's a great question. They actually come from all over, right? The first set of students, they tend to cluster because once one of them tells their friends that they're working in my lab, I tend to get a few of them. So I currently have 3 students from the biotechnology program. I also have 2 students from physics who joined. I was teaching the freshman physics 2 course in the spring, and a couple of those students have asked to join my lab as well.
Terrific.
I would like to ask the following question.
Sure.
How does the enzyme have the directional reaction to enable it to move? When you just look at the molecular mass that constitutes an enzyme, it's not exactly obvious that it has an input and an output that will direct it.
Yeah, that's a great question. In fact, nothing in biology is really perfectly symmetric, right? We all have a left and a right and a front and a back, and the enzyme is no different. The enzyme that I showed is a triangle like this, and in fact, it's because it's a trimer. So it's actually got 3 pieces, so it's a little more symmetric. But each one of those pieces has the ability to take in the substrate and then release the product, or take in the fuel and release the output. And so you're right that it's actually much more symmetric, and then which is why we're going to harness it by putting it onto a very asymmetric DNA origami molecule. That will help give us that directionality. That's the other reason why it actually was still just diffusing, but diffusing faster, because it not only doesn't really have a great directionality within an individual enzyme, but it also rotates really quickly. So even if it gives it a push in one direction, it's not very much time before it rotates, before it gets the next push. And so your question is incredibly astute. It's exactly right on. And that's the reason why we need to harness it and actually attach it to another molecule to get it to be more like a rocket.
May I just have a follow-up? Oh, hold on. Let me see. Mute. Right.
No, go ahead. That's great. Okay. Jenny, David had a question. What might be the time frame for the self-repairing infrastructure you referred to?
Oh my gosh, that is such a good question. There are actually engineers who are working directly on this. They're trying to, right now, again, thinking in this bio-inspired way, a lot of people are trying to do things like, they're actually inserting bacteria into the road. So when they crack, there's actually a bacterium that gets exposed and it has the ability to create new material. So again, you're thinking about that cell division process and how do we create new material. There are people who are working on vasculature, so roads that basically have veins to allow the deposit of new material. And then my side of things coming from that more, you could call it more theoretical, we're very much interested in like kind of the basis of this. How would you even power something like this? How would you create a completely new material de novo? So a lot of people who are already working on self-healing infrastructure, they're starting from asphalt or they're starting from concrete and thinking about how to modify it. So they're kind of top down. We're very bottom up. We really are trying to figure out how we could create a brand new material. And ultimately, when we synthesize this, what we're gonna do is not use protein enzymes at all. We're gonna create new chemicals and new molecules that can serve as the basis, but have the similar characteristics that the proteins have. But as far as timescale, I wouldn't want to give you an exact timescale, but I would love to see a lot of progress in this in the next 15 years.
Jenny, another question says, we saw experiments with energy. Are there any environmental applications that have been the subject of the experiments?
Yeah, so one of the other like big Imagine slides that I tend to use, but I didn't use here was, this could be used to help with either remediation of our current environment, so cleaning up the environment, or terraforming of other planets. One of the nice things about having systems that are autonomous is that they don't need your help. Something that actually might be a little bit closer than 30 or 40 years out for maybe some of these remediation things would be something like in California when they have forest fires, they often have mudslides that follow immediately after. Creating new materials that are inert to the environment except they also are difficult to slide, would be something that you could envision, or maybe that you could just kind of lay down and plants just grow around them, all kinds of stuff. They're fire resistant, but then they're also kind of change their viscosity property. So maybe they're just little grains, but as they start to feel a lot of movement, they can stick to each other. There are people who are actually very interested in sand piles, how sand moves, how sand and other granular materials are actually able to move. In fact, Lisa Manning, who's the leader of Bioinspired, one of the parts of her research is thinking about how these types of materials can change their properties over time. And if we made those materials active, where they could move themselves, they could actually work to hinder a mudslide or hinder a landslide like that.
There was a question, Randy asked, can you say a little bit about how you get the DNA for the form rod shapes? And also, are you embedding substrate or enzymes into the DNA?
That's a very good question. So we get the DNA from a company, we just buy it. It's actually a pretty crazy way to do it. So we take one very big round piece of DNA that would normally be in a bacterium, and that's actually the basis that we use for everything. That sequence doesn't change. But what we change are what we call the hairpins that are in between. So we actually have these little pieces of DNA, and they work to kind of cross-link between other two parts of the big round piece of DNA. And so it's weird because it's like this big circle of DNA that actually gets folded up and crunched with all of these like thousands of little tiny hairpins that are in between. In order to use that as the kind of carriage that we will harness our enzymes on, we are specifically binding the enzymes to that DNA. There is a small molecule chemical that you can bind to the ends of enzymes that will allow you to stick a tiny piece of DNA onto the enzyme. So we're creating kind of a chimera. We've got an enzyme with a little piece of DNA hanging off. And now we can use the sequence specificity of the DNA to hook that to another piece of DNA and then on the rod. And so that's the chemistry of how we're binding everything together. The fuel is actually just free in solution. And we have a novel microfluidic device, actually, which I could go into more if you're interested, that allows us to exchange the small molecules. So the fuel we can basically remove the product, we can remove all of the exhaust, because that can inhibit the enzyme reaction. So we want to get rid of that and then constantly be giving it fuel. And so that will allow us to perform these experiments over long times as well.
Okay, two questions about enzymes. One from Alona, what kind of enzymes do you use for catalysis? And can we correlate this information to the mature X-ray crystallography body of information about enzyme kinetics?
These are great questions. I feel like I'm at a real seminar here. You're all going to be like, she didn't explain any science. I have explained enough science. I will say that the first enzyme that we're using for this is urease. It's not, it's because it's a very fast enzyme. It turns over very quickly. It's also one of the first enzymes that was ever, where it was ever shown that you should be able to get faster diffusion from the enzyme. And it is not found everywhere. It's not found in every cell type, which actually suits us just fine, because if we ultimately want to test this with other biologicals, we actually want things that are kind of orthogonal to each other. We want to add something that doesn't really exist into the system that isn't going to interfere with other aspects of the system. So actually that works really well for us. Like I said, the enzyme is urease that we're using. It has been shown that other enzymes can have similar activities where they go faster. It does seem like the enzyme needs to be exothermic, that seems to help. So it's possible that there's some temperature dependence of these things. Again, if you're thinking about what's the theory behind this that destroyed Einstein's theory, these are some of the things that people are looking at, but we don't know for sure. Although it's been shown that there's an endothermic enzyme that can do the same thing. There was a second part of the question, but I missed that.
Yeah, the second part of the question, can we correlate the information to the mature X-ray crystallography body of information about enzyme kinetics?
Yeah, that's a great question. So this is where a lot of the theorists are trying to really understand how this is happening. One idea is that, okay, so maybe you have a triangle, but maybe this thing is actually changing shape, right? And that it's not that it's like it's a swimmer. I don't think nobody thinks it's going to swim around, but that just changing the shape in this direction would actually allow you to diffuse in this way faster and diffuse in this way slower. And so maybe the increase in the diffusion is actually not due to it being like a rocket. And so that is actually one of the questions we will be testing when we harness these enzymes onto, right? So if that is the case, then it won't actually propel our rocket at all. And so this is actually a big open question that we'll be able to test by trying to harness it. So for instance, if horses were just actually like tumbleweeds and they just really moved, but they just randomly moved, right? And then you tried to harness them, your wagon wouldn't actually go anywhere. So we need to figure out, is our enzyme more like a horse or is it more like a tumbleweed? It's like something that can move, but it's not really moving under its own power.
Okay, Kevin, you're up next. Go ahead and unmute yourself and ask your question.
Thank you so much. It was a great presentation. And in terms of the Terminator analogy, do you have to worry about having different substances choreographed together, working together, or having one substance like the liquid metal doing different jobs? Because there's like two different aspects of having many things working together and then the same thing doing many things.
Yeah, exactly. So again, thinking back to this being inspired by biology, biology is heterogeneous. And it really does have all of these interacting players that are interacting and working with each other. And ultimately, I think that's what we will need. We'll need a material that's a composite material. And composite materials, I mean, many of you are interacting with composite materials all the time now, right? Because they do give materials extra enhanced properties, better strength, less fragility, all of these things. And so it wouldn't surprise me if we ultimately need to create composite materials in order to achieve a lot of these things. So even just the idea of attaching the protein to DNA origami, we're already starting to think about that. But I think what you were thinking is maybe there's more than one system. There's the response system, the sense system, and then those will have to talk to each other. A lot of people in biology are looking at these things. And in fact, they're dissecting them again from the top down, thinking about synthetic biology, like how do the different parts of the cell talk to each other? And can we then replicate those or program them ourselves? And so I think actually, even though I'm not a synthetic biologist, a lot of what we're trying to do actually fits with synthetic biology because of that. So that's what synthetic biology means. So that's partly why I use that word synthesis, because taking into account the idea of the human made or the human altered part, right, where we're trying to do that.
Just one follow up, I'm sorry, is are you working on seeing what works, or are you in your mind saying, let's see if we can make this work? Are you doing a little of both, or are you kind of seeing just how whatever happens, happens?
I think we do a little of both. I am really enamored with discovery-driven research. I like it because it's if it fails, you've still figured something out, right? And so and actually, it's really great for undergrads to do discovery-driven research because they don't feel the pressure of feeling like I have to prove a theory. They don't ever have to prove anything. We're just trying to figure out how the world works. And so what we do is what I do a lot of times is I'll give individual students just their little question, hey, if we change the salt concentration, what happens to this thing? And so they each have their own little part. And one of the ways that we've been so successful at being able to get more of those students as published authors as undergrads is that each one of those little parts becomes a figure in a bigger paper. And so we've had many papers in our labs that have been driven by kind of a team of undergraduates, each working on their own independent projects, but still working as a team in tandem to create new information. And they don't ever feel that pressure of that they have to get something to work or it has to work, because whatever nature tells us is the answer.
Excellent. Thank you so much. Thank you.
Wow.
Stephen had a question. Which Einstein principle did you disprove or was disproved?
So it's often called the Stokes-Einstein theory. And it's basically says that the diffusion coefficient depends on the temperature. So it directly depends on the temperature. So if you make something hotter, it should move faster by diffusion. And it inversely depends on the size of the molecule and the viscosity of the water or whatever it's in. And so what we've done, and the reason why we disproved it, and again, I didn't go into a lot of this, but if you read that paper from Physical Review Letters, we did some back of the envelope calculations on temperature. We didn't really specifically, we didn't alter the temperature because the idea would be that the enzyme might be changing the temperature locally, and that's why you would get faster diffusion. But if you do a back of the envelope calculation, it should dissipate within 10 to the minus 12 seconds. And it should only be able to go out like 1 atom away. So this idea that like this very macroscopic, heat will change is actually doesn't seem to be true in our system, that the heat there's no real heat transfer there. So that's the numerator. And then the denominator, you could think maybe you change the viscosity, but it doesn't seem like, again, the same kind of idea as the temperature change. And then the other option would be the size. And this is one strength of the single molecule imaging that we use, the fact that we can see single enzymes, is we can use the intensity of the fluorescence to actually tell you how big the molecule is. And so we can't really tell you exactly the shape. Like I can't tell you if it's like this or like this using just the method I used just now, but I can tell you if it's 1 versus 2 versus 3. And so by using that quantitative fluorescence imaging, by using our single molecule imaging, we were able to count basically how many of the individual proteins were coming together to make one of these protein units. And we actually saw no difference whether or not there was fuel present, implying that the size wasn't really changing when it was turning over its fuel.
Great. We've got a few more questions in the chat. I want to go to Don right now. Don has his hand up. Go ahead.
There you go. Good to see you again. It's been a while since we had lunch. What was it, November, I guess, at Syracuse? And I was fascinated.
Yeah, great to see you.
And I was fascinated with the topic at that time. And I've been wondering, since then, and with your presentation today, whether you believe that there should be more collaboration potentially with either other institutions or other folks, since this technology, I would think in theory, has a lot of promise, but it may take a while to get there, and maybe with a collaborative nature, we could move faster. What do you think about that?
Yeah, so as I said, this new grant that's funded from the NSF for this work, where we're harnessing the enzymes, is actually being funded by the NSF, and it's collaborative, right? It's actually a collaboration with 2 other experts thinking about these topics. One person who really thinks about more of I would say the statistical nature of this, like how do you have many of these together and then harness all of them? And then another person who's really an expert on how he's more of a manufacturing guy. Like he's like, how do you actually make these little particles the way that you want them and control them? And then I'm kind of the enzyme, biochemistry, biophysics kind of expert. And so the 3 of us together are going to continue this work. As far as larger scale, like how do we create teams larger scale? I think the Bioinspired Institute is absolutely one of the places where we're doing this. And so I'm part of a couple of different pieces of that institute. One is called Smart Materials. And so I think you can see based on my presentation where I fit in there. Can we make smart materials of the future? But there's another part which is more thinking about the opposite problem. What do we learn from biology by studying these things? And so that one is more on mechanics of material, of cellular systems, and looking, and in particular in tissues, building up that way. And there's a whole brand new group that's just formed this year I'm really excited about. They're called Form and Function. And the name is maybe a little bit vague, but they're really thinking about large scale things. How do whales move in water? right? How do snakes slither in sand? And what can we learn from that in order to help with these principles? So I'm really excited to be interacting with that group of researchers too, because that to me is going to get to these questions of the infrastructure, the remediation of our environment and things like this. We're going to be, and then even things like creating new prosthetics, right? So that kind of getting that medical aspect in there too. And so for me, I'm really excited about that new group. And so I think right here in our home base at Syracuse University, we actually have a lot of people interested in these ideas and moving this forward. And then of course, we all also have collaborations outside that we're bringing to bear on a lot of these really cool questions.
Okay, thank you.
Okay, a couple more questions. Richard had a question about the smart materials. He says, if clothing could protect people from viruses, et cetera, would the clothing be able to be reprogrammed once a new virus evolves, i.e. corona? Would the clothing need to be reprogrammed like a software update? Could it be hacked, et cetera?
Yeah, that's a great question. The way I've been thinking about this is, so you could think about it as like having a specific detector, right? Like your antibodies do. We all know now everybody's an expert on antibodies and things like this now because of the news. Or you could think of it as just being a size filter. So if it's a size filter, I mean, viruses are really small, so it's going to have to be a small filter. A lot of these filters like the N95 masks, they actually work on electrostatic interactions. So it's not so much that they have very, very tiny pores. It's just that around the pore is something that is kind of attractive to the virus. And so changing the electrostatic nature of your clothes is something you could do, right? And in fact, it happens naturally a lot of times. If you're ever wearing like a silk blouse or, a silk shirt, I don't know if many of you men are wearing the Seinfeld puffy pirate shirt, but if you were and it was made of silk, you'll notice in the winter and you wear one of these and it's like it starts clinging to you, right? Because it's like picked up all these electrons. And so you might be able to create materials that could alter their electrostatic properties, which would then, when the virus was around, it would like kind of suck it up, right? And so the virus would be on your clothes and not on you. And so that would be a way to do it. In fact, that is, again, like I said, a lot of how the N95 masks work.
Question on a follow-up to the DNA question from earlier. He says, I assume that with PCR or CRSPR, you could create DNA sequences for improved efficiency of whatever action is your focus.
Yes, you can. And I mean, one of the nice things about it actually is you could like kind of slightly modify and evolve. You could actually use an organism to help you evolve these slight pieces of DNA. And you could just do a screen where you just screen a whole bunch of them and see which ones are the most efficient. The person that we're working with, who's, he's really the DNA origami expert on our team, he does some stuff like that, and he's much more into that field than we are. I just want to apply it and I want to make a rocket ship out of it. But you absolutely can do things like that. And people are working on how to make DNA origami folding more efficient. You might think that using DNA as a particle would be very expensive, but it actually turns out there's a lot of DNA in the world. There's a lot of it. There's a lot of it just in your own body. And so, and it's actually fairly easy to get if you can exploit other animals, not animals, but bacteria and things like that to make it for you. And so, so it actually is a lot easier to get a hold of than it might have seemed at first when these technologies were first kind of discussed. And DNA origami has really taken off in that way.
There's a comment, we are looking to use Nano Septic as a contact material to oxidize virus on contact. And he or she says this for campus private housing would be great for COVID mitigation when students return.
I don't know if we're doing that. That sounds like a really great idea. That sounds fantastic.
Yeah, we'll have to learn more about that. I think that is the questions that we have. Unless there's any more here, we have one from, are you planning to use DNA rocket concept to quote unquote heal infrastructure?
Right. So I think ultimately we would probably want to make a synthetic version of that before we put it into infrastructure. And so what I mean by that is instead of using amino acids that are turned into proteins, right? A string of amino acids, that's what's in the protein, that's going to degrade over time very quickly. So proteins don't really last outside of your body for very long. Is a good thing. But so we would, I think what we would want to do is work with our chemistry colleagues and actually create molecules that are, maybe polymer based. So polymer and even could maybe even fold into the right shape, maybe even integrating, they can make all these kind of amazing polymers that are like bottle brushes. So in a sense, maybe all one thing could be polymer based, but one half of the polymer strands could actually have the ability to perform a chemical reaction. So if you remember back to the little beads that had the magnetic thing on them and they could make a little windmill and there were 3 of them spinning around, that's completely synthetic. That's made of plastics and metals. And so I'm thinking what we would do, we need a molecular scale version of that. That is basically a polymer. Polymers are often the basis for plastics, and so that's how this would be. People are working on self-healing plastics that work in a different way, where when they rip, they can actually reattach to each other because the bonds are not 100% covalent all the time. They actually turn over. They can become covalent, and then they can fall apart, and then they can reattach to a new part. And so these are called vitrimers. And so people are actually looking at that kind of technology and how you would be able to kind of heal plastics. So it may be that in the future, our roads are more plastic and less concrete and less asphalt.
Okay, we got a few more minutes. The person who had the comment about the Nano Septic material wanted to make a comment. Go ahead and unmute yourself. Okay. All right, go ahead. We can't hear you. Okay, I'm going to go ahead and I think we have covered.
Maybe you can hear me. Can you
yeah,
hear me now?
Go ahead. Go ahead.
No, sorry.
Okay.
So anyway, this is still a good read if anybody's looking for something to read. Bleeding Orange by our famed coach Jim Boeheim. Nano Septic technology is something we just discovered last week. And the private housing is in a fraternity housing corporation that I'm an executive board member on. So I would encourage people to look at I think it's nanoseptic.com and just look at how does it work. I think you'd find it very interesting, but they have multiple different substrates and venues for where this kind of double-faced material allows you to, for instance, put your hand or stand on a mat or even mouse pads for crying out loud. That will oxidize viruses, and it's all powered by light. So it seems like it just keeps on going, kind of like the ever-ready battery. But in this day and age, I think it's a good use of that kind of applied technology, analogous to the point of discussion today. So I just wanted to make that comment. Thank you.
Terrific. Thank you. Okay, I've got one more question, and I think this will wrap it up because this seems like a big one. Chris had the question, can it be possible for this procedure to someday be used to create a 4th spatial dimension?
I don't know about this technology. That might be a different physics talk.
We'll find an expert on that and get an event in the queue on that topic. So thank you. That's a great, good prompt. So there's been some very good comments about Professor Ross about your work and how people are impressed and they want to thank you. So I think that's a good way to end this. So I want to thank everybody who participated, and we appreciate your interest and hope you enjoyed this. We're going to be doing some more of these on science topics, humanities, all kinds of different things. So keep an eye out. And Professor Ross, thank you so much.